Today we're talking about mitosis and cancer. Now we've already kind of covered a little bit about mitosis, but it does help to review mitosis before going into cancer because the two are very much related. At its essence, cancer is out of control mitosis. So what we mean by that is the process of mitosis is controlled by chemical signaling activities in the body. And when those chemical signaling activities get messed up, then we get out of control mitosis, and that's what cancer is. But cancer isn't just one thing. Cancer can happen with pretty much any origin cell. So there are as many types of cancer as there are types of cells in the body, which is quite a lot. So you are at a point in your biological sciences education where you have all of the tools and all of the cell biology that you need to at least understand cancer. So what we're going to do is review, first of all, the cell cycle, which means that we're going to talk about not just mitosis, but interphase as well. So you may recall that there's going to be G0, which is essentially the cell is going about its business. If it's a stomach cell, it's doing stomach-related activities. If it's a pancreas cell, it's doing pancreas-related activities. That's G0, so to speak. But every once in a while, the cell will get chemical signals that say it is time to divide, and then it's going to start G1, S, and G2 phase. Now, depending on the source you're coming from, G1 sometimes refers to gap, or G sometimes refers to growth. I'm actually a bigger fan of growth phase 1 because it's more descriptive of what's actually happening. So the cell is going to ramp up its metabolic activity. It's going to duplicate the number of organelles. It's going to multiply. It's going to increase its organelles and any cytosolic components. So it needs to grow before it can divide. So if you remember from cell bio lecture, I like to talk about a sort of a factory metaphor for the cell. So in this case, it would be like, okay, we're going to open a new factory, so we need to make twice as many factory equipment, make sure that we have twice as much as we usually have before we open this new building. So that's comparable to what's happening in G1 phase. Now, something that's not on this chart, but would be really useful here, is that in both G1 and G2, you're going to have something called checkpoints. So we are going to check the genetic material. We're going to check the material in the cell for any mutations, both before DNA are replicated and after DNA are replicated. So both G1 and G2 have a checkpoint to check for mutations in the DNA. Now, if there are substantial mutations detected, then there are cascades that will occur that lead to apoptosis. And that's actually going to be a key idea in terms of how cancer is regulated in the body, how we actually have some really specific mechanisms in the cell to initiate apoptosis. And then apoptosis is a self-destruct button. It's a self-destruct of the cell. It's like an implosion of the cell. So like the lytic enzymes eat the cell from the inside. And it's, in some ways, I don't want to say good for the body. We can have apoptosis in a bad way, but in terms of preventing cancer, it's beneficial. And then one cool thing about apoptosis is it does not cause inflammation, whereas necrosis, one day you will learn necrosis does cause inflammation, and inflammation has its own kind of harm in the human body. So apoptosis isn't harming the human body any more than just destroying the single cell. And now that we've had enough histology and we've looked at tissues quite a bit, hopefully you have the concept that there are many cells, right, in any given tissue. So G1, we are increasing the size of the cell, we're increasing the organelles, and we also have a checkpoint to check for mutations. If mutations are formed, we will initiate apoptosis. Apoptosis is programmed cell destruction and self-destruction, and it does not cause inflammation. So it really is just there to prevent cancer. So after G1, growth phase 1 or gap phase 1, that cell will go into S phase, which I generally say stands for synthesis phase. So we are going to undergo DNA synthesis and replication at this point. So we've already had our DNA synthesis lecture, so we already know all of the enzymes involved in that process, your helisase, your DNA polymerase, all of that thing. So we should end up with 2 copies of each chromosome connected together as a sister chromatid after S phase. As 2 sister chromatids after S phase. And then we go into G2 phase, which is really similar to G1. This cell continues to grow. We synthesize more proteins, more enzymes. Again, we're just doubling the content of the cell. And like I've said, there is another checkpoint in G2. So now that we've copied that genetic material, we want to detect if there were copy errors. So we wanted to check it before we copied it, and now we want to check it again after we're copying it, so that we know that the thing we're copying doesn't have errors, and we also know that the thing we copied didn't create any new errors. And in either G1 or G2, if mutations are detected, the cell can initiate apoptosis, which is going to be generally beneficial, generally is going to prevent tumors or neoplasias from growing. Now, assume all of this works perfectly. We get through G1 S phase and G2 phase. We get through both the checkpoints and no mutations, or at least overwhelming mutations, are discovered. Then, at the end of interphase, we'll go into the mitotic phase, or M phase. And so we have our standard stages: mitosis, prophase, metaphase, anaphase, telophase at that point. And we will have one cell that is going to divide and become 2 genetically identical daughter cells. Now, they should be genetically identical. We've already kind of covered the concept of genetic mutations can occur that would not be detected by those checkpoints. Obviously, people do get cancer, so we know, and we have this idea that yes, mutations can get past these checkpoints. It is possible for that to happen. And genetic mutations or changes might have happened somewhere in here. Less likely, but most likely it happened here. So there you go. You will have cytokinesis at the end of M phase, which brings you back into G0. And again, if you get signals to divide, then it'll continue on into G1 once again. Now that's, again, depends on the rate of cell division in that tissue. And that varies a lot. So in your skin, at your stratum basale, for example, you have mitosis all the time in that stratum basale. It's the stratum germinativum is the other term for that. It's always germinating new cells, right? So there's constant mitosis in a cell like that. Or remember the onion root tip mitosis images that maybe you saw in high school. It's a growing onion root. It's growing into the soil. That's growth. So anywhere where there's rapid growth, anywhere where there's healing in the adult, that's going to see a lot of mitotic activity. But there are tissues in adults that are non-mitotic. So for example, muscle tissue, skeletal muscle, cardiac muscle, these are non-mitotic tissues in the adult. You've got all of your muscle cells. They do not replace themselves via mitosis. Once you injure a skeletal muscle cell, it does not heal itself via mitosis. It gets replaced by connective tissue. So if you injure a skeletal muscle, you injure your biceps brachii, and it gets replaced by connective tissue, you have less contractile tissue at the end of that injury. And that's true for your heart, too. So if there's apoptosis on your heart muscle or necrosis of your heart muscle and those cells die, they don't get replaced by new cardiomyocytes, cardiac muscle cells. Those do not undergo mitosis to replace themselves. If anything, you get scar tissue, which is a type of connective tissue, and you lose contractile ability of that heart muscle. It becomes less effective. And that's what a heart attack is. It's called a myocardial infarction. There's a lack of blood flow to the muscle cells of the heart, and they die, and the heart muscle becomes less effective. So whether a tissue is mitotic or non-mitotic has a lot of clinical significance in terms of healing. It also has clinical significance in terms of cancer, because if a cell is not naturally mitotic in a mature adult, it's actually less likely to become cancerous. So we'll go forward and we'll talk about why that is but I want you to keep that in the back of your mind while we go through this. So again, you have your phases of mitosis. Prophase is when the mitotic spindle forms. We have centrioles migrating to the cell's poles. The chromosomes condense. In the next slide, we're talking about what I mean by chromosomes condense. We go from loose spaghetti chromatin, like loose DNA, to condensed DNA, which is in the form of chromosomes. And then the nuclear membrane dissolves, so you'll no longer have a true outline of a nucleus, and the metabolic activity decreases. So for metaphase, we will have that metaphase plate. We will have an equator between the centriole poles, and I always think that's really obvious to look at when you're looking for examples of mitosis in a cell. Anaphase is when the sister chromatids are actually going to separate away. So we will have an elongated cell with the sister chromatids being pulled to either side. So instead of having a single line, you're going to have two lines of sister chromatids. And then in telophase, it's essentially the reverse of prophase. We're going to reform the nuclear envelope. We're going to have a cleavage furrow that's physically starting to separate what are going to be two different cells. So you're going to have all of your chromosomes collected in two locations, one in each future daughter cell. Duplication of centrioles. And then finally cytokinesis is when that cleavage furrow splits right down the middle and separates the cells into two identical daughter cells, which again, this is mitosis, so they're diploid. They should have the same number of chromosomes as the cell that gave rise to them. Here's what I mean by chromosomes condensing. So you start with a loose strand of DNA. So it's just the DNA double helix, loose, like spaghetti on a plate. You would see it inside of the nucleus if you're just looking at the histology. And we're actually going to use a protein called histones, and we're going to wrap that DNA double helix around histones. And then that creates a complex, which can then be wound again. So you have something that gets wound up, and then you have something that gets wound up around what was wound, and then you wind that up. If you're old enough to have had a telephone with wires, I'm old enough to have had telephones with wires. You know, they had that little spiral going on, and sometimes if you spiraled them too much, they would curl around each other like that, and you could just keep doing that and doing that. So at the end of this, if you actually take a single strand of DNA from any one nucleus, and you were to unravel it, it would be 6 feet of information. It would be 6-foot-long DNA double helix for every single chromosome in every single cell in your body. So one cell, 46 chromosomes. Every one of those 46 is going to be about 6 feet long. So that gives you a concept of how much DNA we have, and then how effective this winding is to condense it into a chromosome. That is a lot of winding. Winding upon winding upon winding gets us a very condensed object, and that's the chromosome. So it's actually 2 copies of one chromosome at this point held together at a centromere, and that's the thing that's going to get pulled apart during mitosis. So here they are visually again. So again, if I'm testing you on this, it's visual, it's verbal. I could use images from something like this PowerPoint, or I could use verbal descriptions for each of the stages of mitosis. So how's it going to look in histology? How's it going to look in an animation? And what's the description of what's going on? But we've already had that, so I'm not going to go over it again right this moment. We can just go back to the cell, this slide right here, and use it to describe those images right there. Now, if we're going to start talking about cancer, we need to start talking about the things that control the cell cycle. So one thing that controls the cell cycle is transcription factors, which regulate gene expression. So they have a lot of ways. What I mean by gene expression, remember that genes determine the type and number of proteins in a cell, and proteins have all kinds of roles in the cell. So for example, they might be a type of receptor, or they might be some kind of signaling molecule, or like a receptor for a signaling molecule. And you can increase the number of those proteins, or if the DNA mutates, you can change the shape of that protein. And as you know, the shape of a protein is going to give it its function. So if you change the shape, you change the function. All right, so a lot of transcription factors are going to determine a lot of steps of the cell cycle. Can we have replication of genetic material? Can we have growth and doubling of the organelles? Can we have all of the steps that it takes to create a round of mitosis? And that's determined by transcription factors. And so that's the starting point, transcription factors, but we're also going to pay attention to, again, the signaling molecule itself. We're going to pay attention to the receptors for those signaling molecules, and we're going to look at the specific genes that code for those specific proteins and how those can get mutated and how they can go wrong. Now, one piece of terminology I want to get straight, because we have started talking about cancer now, is what is cancer? What is a tumor? And in fact, there's a word that should be in here, but is not yet. By the time this is uploaded, you'll have it. Neoplasia. So neoplasia means new growth. Let's go ahead and go back to, not animation, slideshow, current slide. There we go, new growth. So a tumor can be a neoplasia, a cancer can be a neoplasia. A tumor might be cancer. A tumor might be malignant, but it also might not be. So let's make sure we have all of the terms. Anything that is a new growth is a neoplasia. That's what that word means. Neo for new, plasia for growth. Tumors may be benign or they may be malignant, which is to say you can have growth inside of a space in your body. You could have a tumor growing on your skin, and it could be completely benign. It may just be sitting there, not changing anything else in your body, not spreading to other parts of your body, not ruining your life in any way, shape, or form. That would be a benign tumor. Once cells of a tumor start to migrate, start to change, start to metastasize and go elsewhere in the body, we consider those to be malignant. Now, if it is malignant, it is cancer. Not all tumors are cancer. A benign tumor is not cancer. I've even heard healthcare professionals say, I had a benign cancer removed. And that is incorrect. Even when healthcare professionals say they had a benign cancer, they did not have a benign cancer. There is no such thing as a benign cancer, because the definition of cancer is that it is malignant. The definition of malignant is that it has metastasized and is growing in other parts of the body. So that's the difference between benign and malignant. There are going to be some histological differences too. So we can, pathohistologists can look at the histology of a biopsy. So you take a tumor, you biopsy, you take a sample, you cut it into a very, very thin sample, you make a slide out of it, and you take a look at it at the cellular level, and they'll see some differences. So they'll see differences in how many cells are undergoing mitosis. They'll see whether those cells resemble the cells that usually live in that tissue. So if you're looking in the skin, you're looking for keratinocytes, and you're looking for keratinocytes that look pretty much like other keratinocytes, maybe some very small changes. Versus if it's a malignant skin tumor, then it's going to be a keratinocyte that looks very, very different from all of the keratinocytes around it. We could have an abnormal size of the nucleus, abnormal shape to the cell. Some other characteristics can be abnormal. The degree of organization is different if it's benign versus malignant. So these are, it'll be more organized and more similar to the tissue around it if it's benign. If it's malignant, it'll be very, very different from the tissue around it. So that's what pathohistologists are going to do. These are all histological differences. There are some other differences too. If it's benign, it may be encapsulated. I think encapsulation is an important trait to consider when looking to see if something is benign or malignant. If there's a connective tissue sheath around it that's really solid, that's actually going to make it harder for that thing to metastasize. So if you are in a type of tissue that has a dense connective tissue capsule around it, and those cells look similar but not identical to the other cells around it, you've probably got a benign tumor, and you may not need to worry initially about physically removing it. You won't have to do chemotherapy to have it removed, is what I'm saying. Here's an example of what I mean by that histology. So this is a smear. This is an epithelial smear. Those are squamous cells. So that might be a cheek swab, or it might be a pap smear. So that, I don't know which one that is. And it doesn't really matter because those are similar cells in both places. So what we have is, here's our plasma membrane for this cell. It's out here. And here's our nucleus, and it's relatively small compared to how much cytoplasm and cytosol there is in this cell. Like all of that, right, is going to be that cytoplasm. There's quite a bit of cytosol compared to the size of the nucleus in this cell right here. If let's say we're doing this as a pap smear, and the pap smear ends up coming back looking like this, we can see that there is, the size of the nucleus has changed, and the ratio of nucleus to cytoplasm has changed. The organization has changed. The relationship of the cells to each other has changed. So this is what we would call dysplasia. This means that the nature of the cells has changed and become disorganized. So if this was a pap smear, we would call this cervical dysplasia, and it indicates cancerous or precancerous condition. That even looks like some mitosis going on right there potentially. So that's really, really active as well. So that's quite a bit different than what you expect to see, and that does indicate some degree of malignancy. Now, we are talking about mutations to genes, specific genes. Not just any mutation can cause cancer. It's kind of like winning the lottery in a bad way. It's entirely random that the mutation would occur on the specific genes that we're going to talk about. So a lot of different carcinogens out in the world, things like cigarette smoking or radiation from even clinical imaging like a CAT scan, those can be considered carcinogens, and that means that those are chemicals capable of causing a mutation. Now the rest of these right here, proto-oncogenes, oncogenes, and tumor suppressor genes, these are targets for mutation. If you mutate a proto-oncogene and it becomes an oncogene, then it's very likely that cell is going to mutate into a form of cancer. If you mutate a tumor suppressor gene, same idea. Once you've mutated that tumor suppressor gene, then that can lead to cancer. So I will go into more detail on what these guys are. So again, a carcinogen is just any potential cancer-causing agent. I do have a slide for some common carcinogens in the environment coming up. But let's start with the concept of oncogenes. These guys are really interesting. So here's what I mean by proto-oncogene. You have proto-oncogenes. I have proto-oncogenes. We all have proto-oncogenes, and they are a normal part of the cell cycle. So I covered this a little bit differently by saying transcription factors, right? This is a related concept to transcription factors. So proto-oncogenes are genes that code for things like growth factors. And a growth factor is a cell signaling molecule that says it's time for you to undergo mitosis. They code for receptors for things like growth factors. Cytoplasmic signaling molecules that say it's time for mitosis. Nuclear transcription factors. It is time to make a certain protein, such as a receptor, such as a signaling molecule. So these are all things that can potentially trigger mitosis, or can increase the rate at which a cell will undergo mitosis. If this growth factor is signaling mitosis, if the receptor receives a signal that signals mitosis, if this cytoplasmic signaling molecule signals mitosis, if this nuclear transcription factor codes for a protein involved in mitosis. So again, we all need these normally because we all have normal mitotic processes. That's what makes them proto-oncogenes. And this is what they normally code for. But if some kind of mutagen or carcinogen gets in there, that proto-oncogene can mutate into something called an oncogene. And we do not all have oncogenes, or at least most cells of our body don't have oncogenes. The dirty secret is we're all developing cancer literally all of the time. Hooray, sleep well tonight. But we have cellular mechanisms that try to destroy any abnormal cells, right? So that's happening all the time too. So something comes in, some kind of mutagen, some kind of virus, some kind of carcinogen comes in and changes the genetic material or the RNA that's about to be translated into a protein, some kind of mutagenic event on the chromosome. And we're going to have what we call amplification of proto-oncogenes. We are going to transform the normal form of the gene, which is the proto-oncogene, into an oncogene that can create cancer. Now, I'm using this phrase really casually right now, onco. What's onco? Oncology. Oncology is the field of medicine where people are treating cancer. So that's where that word came from. So once it's an oncogene, it is a cancer-causing gene. The proto-oncogene is the normal thing. Once mutated, can become something that causes cancer. Do you see how those phrases work? So once a proto-oncogene has transformed into an oncogene, the thing that got made from that genetic material, that protein, changed, and we transcribed and translated a protein that overstimulates the cell cycle, one way or the other, through any of the mechanisms we talked about before. So these were the normals, and now we've transformed them into oncogenes, and all of these are now in their abnormal form as an oncogene. So again, proto-oncogene normal, oncogene abnormal. Let's look again at some ways that proto-oncogenes become oncogenes. One common way for that is to get some form of retrovirus. So you are going to take microbiology pretty soon, and that's going to hit you pretty hard. So we can kind of look ahead to that a little bit. You're going to learn about retroviruses. And what you're going to learn is that these viruses are going to co-opt your cellular mechanisms. They're going to put their genetic material in with your genetic material, and then you're going to go through transcription and translation of the proteins that make up that virus. So in the process of a retrovirus inserting its genetic material into your genetic material, that can cause a lot of mutations, which you would expect. You know, you've got a long strings of A's and T's and C's and G's, and then a retrovirus comes in and puts in a whole different set of A's and C's and T's and G's right in the middle there. That's what happened. So that's an entirely different genetic sequence. That's a mutation, effectively. So retroviruses, they have what we call genetic scissors. There's more specific clinical terms for them. But in doing their process of infecting your cell and co-opting your cellular mechanisms to make more proteins, which are a major component of viruses, all of these specific viruses can then cause cancer. So HIV, for example, was very famous for causing a form of skin cancer called Kaposi sarcoma. Epstein-Barr virus is commonly found in Burkitt's lymphoma, and actually others as well. Human T lymphocyte virus type 1 is implicated in adult T-cell leukemia and lymphoma. So there are a number of viral infections that can lead later on to cancer. This isn't even touching on human papillomavirus. So human papillomavirus, probably not a retrovirus, that's why it's not on the list, causes genetic mutations as well, and that can lead to cervical cancer quite a bit. So just because you had Epstein-Barr at some point doesn't necessarily mean that you're going to end up with Burkitt's lymphoma. I'm not showing you this to make you stress out about that. A lot of people have been infected with Epstein-Barr virus. but we do find the genetic material for these viruses in a lot of cancer cells. And so that's how we've gotten this connection out of it. So again, that's just one mechanism for a variety of cancers, but these are particularly well connected between the viral infection and the cancer. Now we've covered oncogenes. I do like to talk about tumor suppressor genes. These guys are really interesting. So a tumor suppressor gene, again, just like the proto-oncogene was perfectly normal, a tumor suppressor gene is a perfectly normal gene that we all have. So I mentioned G1 and G2, you have checkpoints. Remember those? And during these checkpoints, if a mutation is discovered, the cell will initiate apoptosis. So those would be proteins. The proteins responsible for signaling apoptosis in the event of mutation, those would be coded for by tumor suppressor genes. So if you have functioning, healthy tumor suppressor genes, then during G1 and G2, you have checkpoints. They check for mutations. If mutations are found, we initiate apoptosis. The cell self-destructs and saves all of the tissues around it. Everybody's happy. So if we're going to develop cancer out of a tumor suppressor gene, that means that this gene has to mutate in a way where we do not get the proteins from that genetic material. Okay, so we have a mutation to a tumor suppressor gene, then we transcribe and then translate that into a new protein, but this protein is unable to inhibit the cell cycle. It cannot stop mitosis from occurring. It cannot promote apoptosis in the event of mutations being found. It's just a defective, ineffective protein because we mutated that one. So it's totally random that these are the ones that got mutated. It's totally random when it's a tumor suppressor gene. It's totally random when a proto-oncogene transforms into an oncogene. It's just like winning the lottery. It's incredibly unlikely that that one would get hit, just like someone always wins the lottery eventually, right? Somebody won the lottery. You have millions of cells in your body, so it's very unlikely that any one cell will have that particular gene win, but it's happening a million times over, so eventually it happens. So in carcinogenesis, of course, we're going to start with DNA damage, some kind of mutation, maybe a virus, maybe radiation, maybe some terrible chemical that you got through cigarette smoke. And that leads to initiation. If we have enough growth promoters around, more mitosis promoting molecules around, then we'll go into the promotion phase. And then we will develop a cancerous phenotype, and that's called progression. We'll come back to this for just a second. I want to connect it to the idea of metastasis. Now, where are we? So again, one cell won the lottery, so to speak, and one of these cells is in possession of a genetic mutation that causes out-of-control mitosis. So that cell is dividing more rapidly than normal. We haven't had enough mutations yet for the cancerous phenotype to develop. It has to keep going, actually. So it's already mutated once. They've color-coded it, so we have all these beige, we'll just go with beige, beige epithelial cells here, and it's just had one mutation that causes extra mitosis, and now color-coded in purple is these cells have mutated again. They're changing form. They're changing form into something that can metastasize. They have to develop certain skills, so to speak. They're not people. They don't have a will. They're not actively malicious. As much as cancer seems like this malicious thing, it's just random cellular events. So if these guys won the lottery once, these guys won the lottery twice, and just happen to have mutations that lead to growth or the ability to degrade this basement membrane and make it into the underlying connective tissue. Or maybe it can change into a form, mutate into a form of a cell that can survive the forces in the cardiovascular system, because not every cell can survive in the cardiovascular system. Your red blood cells have a really dense cytoskeleton because they're going really fast in that cardiovascular system. And there's a lot of what we call shearing forces in there. So what I mean by that is those cells could just shear apart if they didn't have that cytoskeleton. So if these are going to metastasize, if they're going to go to distant sites and grow there, then they have to be able to survive in the cardiovascular system. Not everything metastasizes via the cardiovascular system. In fact, very few do. You just don't know enough about the lymphatic system for me to be talking about the lymphatic vessels. So there are other vessels that are not quite so high intensity alongside the cardiovascular system. So it's much more likely most tumors are going to travel through the lymphatic system and lymphatic vessels rather than the cardiovascular system. And that means that we need to change fewer aspects of the cell in order to get from one place in the body to another. And we call that, again, metastasis, once we've moved from one part of the body to another. So in this case, let's say that this is your skin, and this is a keratinocyte that was our starting position. More likely would be a melanocyte. Let's say it's a squamous cell carcinoma. So let's say this is a squamous cell carcinoma. This is just a normal cell of your skin that has mutated, become a tumor, become cancerous, and become able to metastasize. Let's say that that keratinocyte ends up in, or that mutated keratinocyte ends up in your liver. It's still skin cancer, even if we took that tumor from your liver. Or breast cancer can metastasize to your brain. So even if we removed a tumor from the brain, it's still breast cancer. So the cancer is named for the origin location. If it originated in the breast tissue, then it's breast cancer. If it originated in the skin, then it's skin cancer. If it originated in the lung, then it's lung cancer. Now again, some common carcinogens. Tobacco is a huge one. There are still conspiracy theories out there that say, oh, you can get lung cancer without tobacco, so tobacco doesn't cause cancer. Yeah, tobacco definitely causes cancer, very, very much so. Diesel exhaust, benzene, that can be found in certain paints, dyes, furniture, finishes. It's pretty flammable. If I remember that correctly. Pesticides, UV light, UV radiation, like from the sun. I say that, but it's pretty cloudy today. But you know what? UV comes through clouds. You should still wear an SPF even if it's a cloudy day. Ionizing radiation from radioactive materials, medical and dental procedures. If you have a X-ray or a CT scan, those are particularly radiating. Human papillomavirus, various STIs such as hepatitis, HIV. Smoke, tar, smoke, soot. Asbestos is a big one. Asbestos was it's a fireproofing thing. It is really great at keeping things from being flammable, but it's pretty bad if you inhale it or if it gets into your body in some other way. And then radon. So radon is a gas that seeps through rock. So if you happen to have a basement or, yeah, everybody should test for radon. I actually paid extra to have this house tested for radon because I'm on the side of a hill and my house is on bedrock and there's a gigantic rock in my basement. So it's very plausible that I would have had gas seeping through the edges of that rock or through the porous nature of the rock itself. It was fine. Our radon test came back pretty normal. But that can absolutely cause lung cancer, which is why it's a common requirement for a lot of inspections. Okay, let's see. Now, I already mentioned the word dysplasia, the abnormal change in cell structure. There's actually a few different words associated with changes to the structure of a tissue. Metaplasia, when it changes from one form to another. Atrophy, hyperplasia. Hyperplasia is just growth in number. Hypertrophy is growth in size of existing cells. Not all of them indicate cancer. I would say out of all of them, metaplasia and dysplasia indicate or hyperplasia as well. Those three indicate a precancerous condition more than any other change in tissue. Now, if we can do things called staging and grading, and we'll get to those in just a bit, if our scientists discover that the malignant cells are only in one place in the body, they have not migrated past whatever membrane, whatever capsule is keeping them in one place, we would refer to that as an in situ cancer. Now, we have determined that it is malignant, it could migrate to other locations in the body, but it has not yet, then it is in situ. An invasive carcinoma means that it has degraded some kind of basement membrane, some kind of capsule that separates it from outside tissues. And metastasis means that these cells have migrated to another location, there we go, and they are growing tumors at that distant location. Now I've added this in here, another trait of tumors as they grow, benign or malignant. You might remember from thinking about the integumentary system, from thinking about skin, you may be aware that the skin is avascular. So there's no blood vessels in the epithelial tissues of the skin. And that means as those layers of skin grow away from that basement membrane, they're growing farther away from an oxygen and nutrient source. And as they grow farther away from an oxygen and nutrient source, they die. The same thing happens as a tumor grows. So you don't naturally have blood vessels in a tumor as it grows, right? Let's say you had a skin cancer growing and there was a tumor growing on the surface of your skin and it's getting larger and larger. It doesn't inherently grow its own blood vessels, so that tumor growing on your skin would be getting even further away from a nutrient source. But it turns out one of these key mutations for tumors to grow is angiogenesis. You can grow new blood vessels, and a tumor that is growing successfully has mutated the ability to signal blood vessels to grow more. So this is an aspect of normal physiology. You do have normal physiological angiogenesis. For example, if you injure yourself, then let's say you got a big old bruise on your arm, so your arm is bruised, you might develop some more blood vessels in that area to help clean up that injury, to replenish the tissues in that injury, to repair that injury. So you may temporarily have some new blood vessels in that area. It's just not typical if you just have a random skin cell that this one would be initiating angiogenesis, right? So there are a lot of tissues that don't normally initiate angiogenesis, but if it's going to grow, if it's going to metastasize, then this is actually going to send signaling molecules to the nearby capillary beds to cause angiogenesis and allow this tumor to grow so that none of these cells is too far away from a nutrient source. So those tumors do get very vascular sometimes, and a more vascular tumor is much more dangerous to remove than an avascular tumor or a poorly vascularized tumor. It is possible to cut into a tumor and have a bleeding problem as a result of that. Pretty sure there's a significant episode of a certain medical drama with that plot line, but I'll leave you guys to guess that one. Trying not to do too much copyright infringement today. Where were we? Okie dokie. Did I skip a bunch? Thought I already did that slide. Didn't I already do that slide? We'll come back to here. So, linguistics for your neoplasms. If it just ends in oma, in fact, I'm going to add this right now. If it just ends in oma without a sarcoma, without a carcinoma, this is probably benign. So you can have, for example, a lipoma, and that's going to be a benign tumor of lipids. It's also known as an adipoma. So adipose tissue is what I'm saying there. So that's just a fatty tumor. And those are pretty much harmless most of the time. If you've got a fatty tumor, if you're feeling your low back and it just feels like there's a fatty lump, you should still have it checked out. More often than not, it's just a lipoma. I've seen a lot of them in my massage therapy days, and my dog has a couple of them too. So if it just ends in oma, that's a benign term, typically. These are like 80% accurate. These are not 100% accurate. If it's a carcinoma or a sarcoma, those are cancers. Again, 90% accurate, 80% accurate. And then you have things like lymphoma and leukemia. So those indicate cancers of the blood cells, with lymphoma meaning blood-related cells landing in lymphatic tissues, like lymph nodes, for example. So carcinomas tend to relate to epithelial tissue. Sarcomas tend to relate to connective tissue. I've heard of some sarcomas coming from skin. Again, consider this to be a 75% to 80% accuracy rate, which helps you predict when you're looking at medical terminology. It helps you predict what a certain disorder means. It's not a 100% accurate thing. So one example of that is, let's see, make sure I get this right, ductal carcinoma in situ, which again, I've been saying situ, in situ, but I listened to an absolute expert on this for 4 hours and she said situ. So everybody has their own pronunciation for those things. So ductal carcinoma in situ is a misnomer. That's actually a benign tumor, even though it has the word carcinoma in it. Part of it is that it's kind of this weird intermediate tumor that even pathologists have a hard time with. So there's reasons when those naming conventions don't work, but you have to learn every single one of them one by one. So good luck. Okay, moving on. There are ways to grade tumors, and there are ways to stage tumors. And these are distinct processes that are done by different healthcare professionals. Now, my predecessor, who taught this before me, really likes to work with the School of Nursing, and it's a really important thing for nurses to learn the difference between grading and staging. And we do find that in the long term, nurses have a hard time telling the difference between grading and staging. Even people graduating from nursing school can't explain the difference between grading and staging, and it's kind of a problem. So we'll start your learning here with just some examples of what these things mean. So grading a tumor is about histology. So a pathohistologist, this is like something a pathologist would do. A pathologist would take that biopsy, make that slide, and look at the characteristics of the cells. And what we're looking for is the degree that it has changed. How similar is it to the cells around it? How different is it from the cells around it? Now remember, from our traits of benign versus malignant cells, that the more different the cell is, the more likely it is to have the capacity to metastasize. Because remember, we need to mutate all of those capabilities if we're going to metastasize. It's like I said, winning the lottery over and over and over again. It's statistically improbable until you're looking at it happening on the scale of billions of cells. It's going to happen to a cell eventually. So again, the pathohistologist is looking at the cells and saying, this one is abnormal, but we still have a lot of normal cells. It's not significantly different. We still have cellular organization. So this is a very low grade change. Versus, I'm looking at this biopsy over here, the cellular organization is gone. These cells are significantly different from the typical cells of this tissue. This has a very advanced grade of malignancy. Okay, so that's a pathohistologist, that's a pathologist looking at a slide, saying that and doing the grading. Whereas if we're going to stage a tumor, we need to determine if that tumor is localized or if it has metastasized, and if so, how far. So that's not the pathologist anymore. That's probably the oncologist, maybe a radiologist working with an oncologist. So staging is about patterns of spread. So again, you can't get that from a histology slide. You can't get that with a single sample. You get that from clinical imaging, like a PET scan, like you see here. And you would stage it. We have our general stage 1 through 4. There are different ways to stage different cancers. So breast cancer staging is very, very specific. Skin cancer staging is very, very specific. They all have different staging titles and requirements that are very, very specialized when you get into specialized tumors. So staging involves the overall size of the tumor, which you may not get from histology. Once it gets to a certain size, we may go up to stage 2. Once it has grown outside of the original tissue, we may call it stage 3. And once we see it metastasizing and growing in other organs, once we have lymph node involvement, we would consider that a stage 4. And this is just a side note, but renal cell carcinoma, which is to say kidney cancer, cancers that originate in cells of the kidneys, they're actually one of the few types of cancer that travels through the cardiovascular system. We say it travels hematologically. That is not really all that common, but kidney cancer happens to like to travel through the bloodstream. Now again, results of staging determine the treatment modality. Can we just remove this kidney to prevent any metastasis, or do we need to do full chemotherapy? I also want to point out Gerota's fascia right here. That kidney is encapsulated, and I mentioned earlier that one trait of benign tumors is that they tend to be encapsulated. So the fact that this has grown beyond a connective tissue encapsulation, that's part of the staging as well. So you should be able to tell me who's doing the staging, how the staging is being done, what's the difference between staging and grading. Also, again, who's doing the grading, how is grading done? Okay, getting into our last cancer-related topics. How do we treat cancer? So, of course, if it's in situ, we can simply do surgical removal of a tumor. If it has metastasized to distant sites, we might need to do radiation therapy or chemotherapy. And both of these concepts are pretty wild. We started with mutation, and that mutation led to out-of-control growth. But as you know, if you mutate a cell far enough, it will die. So when we do radiation and chemotherapy, we are mutating the DNA of a cell until it is too non-functional to exist, and it's going to collapse in on itself. Radiation can be very, very targeted. We can irradiate just the tumor and cause it to die. Chemotherapy tends to be more systemic. So we're trying to find every cancer cell in the body. Now, unfortunately, it's hard to be very, very localized with that. So we're just kind of killing a whole lot of cells and hoping that we get the cancer cells. If the cancer happens to be related to bone marrow, then we can do a bone marrow transplant. And if you're thinking that cancer therapies, chemotherapy, and radiation seem like a bad deal, we are working on some new ways to treat cancer. Now, this was new as of 5 years ago, so I should probably look to see if anything new is popping up. There are some really cool things coming down the pipeline. In fact, not just p53 gene therapy, but gene therapy as a whole. We do find that this gene right here is called p53. It is a type of tumor suppressor gene, which we already talked about. So a tumor suppressor gene is a gene that codes for proteins that initiate apoptosis and suppress the cell cycle when abnormalities are found. We do find that p53 is mutated or absent in about a third of cancers, and that's systemically, any cancer. So of all cancers that have ever been or ever will be, p53 is missing or mutated. So if we could put p53 back into those cells that have mutated, then we could cure 1/3 of cancer. which is a really freaking big deal. But gene therapy is difficult. If you're interested in this, I recommend you look into something called CRISPR. It's just capital CRSPR. There might be an I in there, CRISPR. So we do have ways of editing genes, and we are turning that on to humans and seeing if we can start editing genes in actual humans. It's not ready yet. There's no conspiracy keeping a working cancer treatment from market, not really. I know a lot of people in cancer research, and believe me, if they could get a working cancer therapy on the market today, they absolutely would. Nobody's holding any treatments back. I mentioned angiogenesis. Angiogenesis is needed if a tumor is going to grow because we need oxygen and nutrients. So one type of chemotherapy drug is actually there for angiogenesis inhibition. We're actually going to prevent the growth of new blood vessels, and that's going to limit the size of a tumor. Let's see, immunotherapy, if we can get the immune cells to target the cancer cells a little bit more effectively. You actually already have immune cells that target cancer cells. There's just maybe not enough of them. So let's see if we can get more of those immune cells involved. And so on and so forth. So we are getting more and more targeted as time goes on. In fact, in the last 5 years, pancreatic cancer has gone from a 5% survival rate to a 10% survival rate, which I know specifically because of, let's see, I was comparing different types of pancreatic cancer back when Steve Jobs died. He actually had pancreatic islet cancer, which has a survival rate of 95%. Whereas Alex Trebek has pancreatic acinar cancer, which has a survival rate today of 10%. So when Steve Jobs was battling cancer, pancreatic islet, the least deadly form, a lot of people hear pancreatic cancer, and the more common one is the more deadly one, and that was a 5% survival rate back then. I said that in a really confusing way. Steve Jobs had the easy-to-beat one. He died anyway because he did not do chemotherapy. He only did surgery. Alex Trebek has the bad pancreatic cancer, which now has a 10% survival rate. All right, there are a lot of really common types of cancer. It's a really major killer all over the world. In the United States, a lot of them are really preventable forms of cancer. So skin cancer has 3 major types, ranging in severity, ranging in level of danger, depending on which cell mutated. Breast cancer, mostly biologically female people. Prostate cancer, most common after age 50 in males. Again, breast cancer and prostate cancer, they both have a hormonal component, and that actually makes them very common killers for biologically male and biologically female people. Lung cancer, smoking is the leading risk factor, and the second most likely risk factor is a family member smoking. Cancers of the colon and rectum, and if we detect those early, survival rates are up to 62%. I think earlier today I checked this just in case. I think it might be 63% now. But that is an up-to-date statistic, more or less. If you happen to know your family history, then you may be able to prevent certain types of cancers. Some of that is very controversial for a number of reasons. Be happy to talk to you about it in the discussion forum. If you're getting regular medical screenings, that can catch cancers at a very, very early stage before they get very serious. Learn self-examination techniques. They are not perfect, however. Self-examination techniques, something is better than nothing for sure. For breast cancer detection, actually, it's not just about finding a lump. It's also about seeing a change. It could be a dimple. It could be something that's weeping. There may be like some kind of fluid coming out of an abnormal site of the breast, or an abnormal fluid coming out of the breast, discoloration. So it's actually quite a bit more. And I would say that goes for most forms of tumors that are closer to the surface of the skin. Avoiding direct sunlight, using a strong SPF, avoid tanning salons. Watch diet and weight, sure, but I'm not going to fat shame. If you are overweight, then the best thing to do is live a healthy lifestyle, regardless of what your weight is. Don't smoke. That one I think is pretty obvious. I'm going to come down hard on that one. Why? It's expensive and disgusting, so just don't smoke, and it's also going to give you lung cancer. Drink in moderation, if at all. Every once in a while, the news comes out and says, well, wine's really beneficial for you. Wine's really bad for you. The more and more research we do, really there's no advantage to drinking alcoholic beverages. There's nothing good about it. If you want to get some antioxidants like you would get from wine, grape juice is just as good, or even better, grapes. You don't actually have to get pomegranates to get antioxidants. Grapes and pomegranates are on the same level for antioxidants. The pomegranate companies are charging you about 3 times as much as the grape companies. And also, you should know what an antioxidant is, which I don't think I've actually talked about oxidation quite as I could have. So let me know if you want to know about what antioxidants actually are doing for you, if anything. All right, that's it. Let me know if you have any questions.